This module will cover neurobiology. As you can see, there are 49 slides contained within this presentation. There is a lot of information. Please do not be overwhelmed. You'll also notice that the slides kind of have a lot of information on each slide. The reason why I did this is so that it's easier for you guys to review. It breaks down kind of what is written within each of the textbooks, and it allows you kind of like a reference. So don't be overwhelmed with all the information within each slide. I did that to kind of help you narrow things down, and this can also be used as a reference in the future. So we are going to start off with some basic information with the nerve cell. So the nerve cell, the human brain consists of approximately 100 billion nerve cells or neurons. The proper function of the nervous system depends on the communication between these neurons. All neurons are composed of dendrites, which are long and short branch structures projecting out from the cell body. They receive and conduct information to the cell body, and there can be one or many dendrites on one cell. Dendrite spines, extremely fine projections on the dendrites. They are the common location for synapses. Next is the axon. This is a long fiber that ends in enlarged structures known as terminal boutons. The axon serves to conduct impulses away from the body. Terminal boutons are located at the end of the axon. They contain vesicles that hold neurotransmitters, and they deliver the neurotransmitters to other neurons across a synapse. Amino acids. These are important modules that are delivered to the nerve cells via the bloodstream. They are the actual building blocks for proteins. Each cell needs to manufacture proteins. There are some purposes, the purposes being the production of the receptors, the enzymes that regulate cellular biochemical processes, growth factors, and structural elements of the cell. Each cell produces its own neurotransmitters, which are known as messenger molecules, which are secreted by the cell, which may influence the functioning of adjacent neurons. Neurotransmitters are often referred to as first messenger molecules, so you might see that terminology in your textbook. This is just a very basic illustration, and this is actually found within your textbook, of the structure of a neuron. It shows you the cell body, the axon, the terminal bouton, and then the dendrites. And down here, these little circles that you can see within the terminal bouton, these are the vesicles that hold the neurotransmitters. Next is another visual aid to help you kind of understand how information is, how it progresses through and how the neurons communicate with each other. So you can see over here that you have the collecting the information, where the information is integrated, and then down here would be the sending. So you can see the cell body, you can see the axon, and it shows you the flow of the information. It's just another visual to kind of help those who are more visual learners kind of understand the whole process. This is an illustration that is more attuned for my visual preferences. So as you can see here, there is the cell body. You will see the dendrites up here, which receives the messages from the other cells. And this long structure here is your axon. And then it talks, I'm sure you can remember back into maybe a biology class that you may have had in college or even high school, I suppose, the myelin sheath. This is these are these little, they look like sacs, but what it does is it actually covers the axon, and it allows neural impulses to speed up because it jumps in between the nodes, which are the spaces between. And this is just a more in-depth visual aid for those that are having some difficulty. You can see the nodes of Ranvier that I was talking about, where the information jumps. So it kind of hops into those red parts as it travels down the axon. And then here is more of what we'll be talking about here in the future. You will see this is the synaptic cleft. And right here is the receptors. These structures right here are your vesicles, and within those are your neurotransmitters. When cells are activated, they produce a nerve impulse, which results in a surge of electrochemical energy, which begins in the cell body, and then it's passed along the axon, which is away from the cell body, which ends in the terminal bouton, which we saw in that last picture. The impulse causes the release of a neurotransmitter into the tiny space between the nerve cells known as a synapse. And the neurotransmitter can cross the synapse between the terminal bouton of one neuron and a dendrite of another neuron, or between a terminal bouton of one neuron and the soma of another neuron. Communication between nerve cells requires the actions of neurotransmitting molecules that deliver stimulation, or also known as information, across that synapse. Substances can be classified as endogenous or exogenous. Endogenous are those that are produced within the body. Example of those would be insulin, endorphins, and epinephrine, which you may have heard of is being referred to as adrenaline. And the opposite of that is those that are produced outside of the body. This includes medication, herbs, caffeine, and vitamins. Okay, next is the slide with all the terminology needed to move forward in this PowerPoint presentation. There's a lot of information on here. Please don't be overwhelmed with it. I put so much text in the slides as a reminder just to help you guys go back to it. And it really breaks down and it talks about the major points within what is all covered in all three of those textbooks. So I kind of summarized it and provided these PowerPoint slides as more of a reference point for you, because it's a lot of information to retain. So there's no way you're going to be able to retain this just by listening to it. So I wrote them all out so that you can refer back to it. So depolarization is a decrease in potential. So it's when the inside membrane becomes more positive. Hyperpolarization increases the potential. So there are more potassium ions on the outside than there are sodium ions on the inside. And that's because of how the potassium and sodium ions are electrically charged, positive or negative, which we'll get to. This occurs after an action potential, and it occurs before the neurons return to its resting state. Which brings us to resting potential. This is when there are negative ions on the inside. This gives that neuron the break it needs. Action potential is when positive sodium ions move inside the cell that were originally on the outside. So it moves outside to inside. This begins at the axon hillock, where the axon exits the soma. It is produced when the neuron's resting potential depolarizes or becomes less negative, or another word to word that is it becomes more positive. Sodium enters. The area is positively charged. Then the positive charge slightly depolarizes the next area, causing it to reach threshold and open the sodium channel. This occurs when the positively charged sodium ion channels open. They stay open for about 1 to 3 milliseconds. Next, it's all or nothing. This concept is it either fires or it does not. It's all or nothing. There is no small action potential. It's full-blown or not at all. Refractory period occurs after the action potential ends. Another action potential does not occur during this time. Absolute refractory period is when the positive potassium channels open, causing positive potassium ions to rapidly exit that neuron, which causes a negative potential. And cannot produce another action potential because the positive sodium channels remain closed. Relative refractory period is the second phase. This is when positively charged sodium channels open and potential remains hyperpolarized, resulting in a greater depolarization. So as you remember, depolarization is a decrease in potential. So that is when the inside of the membrane becomes positive. So that is resulting in that greater depolarization to reach threshold and produce another action potential. All right, so action potential. There is action potential animations and videos that I included in the module, under the module tab under module 1. I strongly recommend that you review those. It's really going to help you understand action potential. So the resting potential of a neuron is negative 70 millivolts. During this time, the neuron is considered to be in a relaxed state. The membrane is considered polarized, and the outside is more negative than the inside. The influx and outflux of ions will make the inside of the neuron more positive or depolarized. The most common chemicals found inside the ions are potassium, which is noted as K+, and ions that are negatively charged. The most chemicals found on the outside in those extracellular spaces are sodium, which are positively charged, and chloride, which are negatively charged. I know, it's a lot. So moving forward is conduction and transmission. So the process of a nerve activity begins with the stimulation of a cell. Ligands are a variety of molecules that may be able to bind to and activate those receptors. This includes things such as neurotransmitters, hormones, and certain drugs. The activation of excitatory receptors on the dendrites or cell body results in an action potential, which is that brief change in electrical potential from the cell's resting states, which we just talked about under that terminology page. So if you need to go back to review it, feel free. The action potential is converted into a nerve impulse as it spreads throughout the dendrites, the cell body, and ultimately down the axon. At the nerve ending, the nerve impulse performs the critical function of causing the release of neurotransmitter chemicals into the synapse. So remember, those neurotransmitters are actually contained within those vesicles. And those vesicles are what's protecting the neurotransmitters from being degraded by enzymes. Neurotransmitters are released via exotosis, which is the secretion of the substance of a cell. So it's a fancy word for here, this is taken directly out of your basic pharmacology book for counselors. So I just want to kind of break this down based on all the information we just talked about. So here you can see your axon, and the electrical impulse is moving to the right, or that electrical impulse can also be referred to as the action potential. Here, these little circles are neurotransmitters, and they are contained with bigger circles, which are called vesicles. The action potential, or that electrical impulse that moved down the axon, causes these vesicles to dock at the presynaptic membrane. Presynaptic membrane is simply, there's a pre and a post. Pre is what is releasing the neurotransmitters, and postsynaptic membrane is the neuron that has the receptors that are receiving that information or neurotransmitters. So calcium ions bind to the presynaptic membrane, causing those vesicles that have the neurotransmitters within to fuse with the membrane. Then the neurotransmitters are released into the synapse. So I briefly talked about the presynaptic neuron, which is the neuron that is releasing the neurotransmitter, and the postsynaptic meaning it's the neuron that's receiving that neurotransmitter. Here is just another illustration for visual learners to try and get an idea of what's taking place during the action potential and the chemical events that are occurring at this time. Here it is again. So this is actually taken straight from your basic pharm book. As you can see, it labels presynaptic and postsynaptic, which I like. It identifies the synaptic cleft, which is that space in between the presynaptic and postsynaptic neurons. You can see the presynaptic terminal bouton. So we've talked about the terminology and the structures in the beginning of this module. And then you will see those tiny black dots are the neurotransmitters. Neurotransmitter molecules are manufactured in the cell body and are then transported down the axon. So we've already seen a visual of that for storage in the vesicles, which are those little tiny containers that we have seen visuals of. While enclosed in the vesicles, the neurotransmitters are protected from the destruction by enzymes that are contained within the fluid. Upon receiving stimulation from the action potential, the vesicles migrate toward that cell membrane, fuse with it, and for a microsecond create a pore, which is a tiny little gateway or space in the membrane in which they're able to discharge or release the contents into the synapse. Receptors are on the postsynaptic membrane. So what are the effects effects of receptor binding. So neurotransmitter molecules, when they're released into the synapse, are capable of binding to specific receptors. So for example, dopamine molecules bind only to dopamine receptors. They do not have a direct impact on other receptors. So think of it as being a lock and a key. The binding of neurotransmitters to receptors is the primary means by which one nerve cell can influence the functioning of other neurons. So you want to keep in mind too that some neurotransmitters are excitatory, so they activate the adjacent neurons. They increase the likelihood that a neuron will fire. And on the opposite end, there are neurotransmitters that are inhibitory, so they impact the nerve cell by reducing excitability. This decreases the likelihood that a neuron will fire. So keeping in mind, so you're thinking, okay, that's easy. We got certain neurotransmitters that are excitatory, so they're going to increase the likelihood that a neuron will fire, and others that are inhibitory. Well, there are some gray, so it's not black and white. There's some gray in there, and what I mean by that is serotonin is inherently neither excitatory or inhibitory. It depends which type of serotonin receptor is activated, and it's overwhelming, and we're not going to get into details about it for the purpose of your role of psychopharm as a counselor, but there are 14 subtypes of serotonin receptors that have been found. So you can think of, okay, just you think serotonin is serotonin, they're all the same. No, there's actually 14 subtypes of that. So neurotransmitter reuptake, and this is important for you guys to know, especially when moving into antidepressants, for example. So when neurotransmitters are released into the synapse, some bind to the receptors, some are chemically destroyed by enzyme, which is found in that extracellular fluid. Many are rapidly reabsorbed into the presynaptic neurons. And the reabsorption is completed by way of a reuptake transporter pump, which is a protein structure within that cell membrane, and it pumps a neurotransmitter back up into the presynaptic neuron. So next is why I wanted to specifically understand that the neurotransmitter reuptake, because some antidepressants act via reuptake inhibition. So that's your SSRI, which is your selective serotonin reuptake inhibitors. So this is when the drug molecules enters the reuptake pore and it blocks the reuptake. So it can't go back up into the cell. So there's more left within that synaptic cleft. So there's more time spent with the neurotransmitter, so serotonin, within that synapse. So it has a greater effect on the postsynaptic neuron. Neurotransmitters will eventually die off and disappear due to enzymes. So that was something that I talked about before. So here is a good visual aid showing this. So you'll see your synaptic bouton right here. You have your vesicles, which contain those little neurotransmitters. So there are some precursor chemicals that translate into neurotransmitters. So like tryptophan is one of those and it transforms into major neurotransmitters, which we will talk about. Here is your synaptic cleft, and here is your postsynaptic neuron with the terminal bouton. So you'll see here that the neurotransmitters are released and then they bind to those receptors. And then over here is inactivation. So they're here's your pump right here. So the neurotransmitters go back up into the presynaptic neuron, or over here they will die off due to enzymes. Receptors are protein molecules that have been produced in the neuron and are embedded in the cell membrane. A portion of the molecule is exposed on the surface of the neuron membrane. The exposed portion is certain binding sites where messenger molecules are able to attach themselves. So messenger molecules, keep in mind, neurotransmitters. They activate the receptor and the other end penetrates into the cell. Cells may have dozens of different receptor subtypes. So example, a dopamine cell may have dopamine receptors, but it also may have serotonin receptors, opiate receptors, etc., allowing it to be influenced by a host of neurotransmitters and hormonal molecules. Most receptors are located on the surface of the cell, although some may be contained within the cell body. So population receptors are significantly influenced by external factors. So this includes stressors or drugs. The average lifespan of a receptor is 12 to 24 hours. After this time, they wear out and are absorbed back into the cell. One way to categorize receptors is from a functional standpoint, based on the end result of the neurotransmitter receptor. The most basic distinction is inhibitory or excitatory. Another way to understand or consider receptors is from a mechanical standpoint. So the majority of receptor activities can be categorized into 5 basic types, which are often called signaling mechanisms. 2 of the most relevant types of signaling mechanisms in neuropsychiatry are ion channel mediated process, or G protein mediated second messenger system, or metatropic. So ionic actions. This takes place when receptors are associated with ion channels. Ion channels are tiny pores on the surface of the nerve cell membranes. When neurotransmitters, drugs, or other ligands bind to these receptors, it can cause the ion channel to open, which allows certain ions to pass through the membrane, such as chloride or sodium, and those molecules are electrically charged. Ionic actions are responsible for rapid and transient changes in nerve cell activity. Most psychiatric medications do not, so I repeat, do not operate via ion receptors, except for minor tranquilizers or benzodiazepine drugs that facilitate the opening of the chloride ion channels. They enter the CNS, so that's your central nervous system, and are then able to produce the rapid onset of actions like tranquilization. So it's an immediate thing. Now on the other end is the metabotropic action. So this is not rapidly occurring. It uses those G proteins to activate ion channels. They involve a gradual change in neuronal functioning that takes place over hours, days, weeks, and months. When ligands bind to these receptors, they operate by activating second messenger systems within the cytoplasm of the cell. The process is said to involve a cascade of chemical actions that ignite a number of intracellular biochemical events. These alterations in the cell's inner chemical microenvironment operate to change or regulate cellular activity by influencing gene expression. All right, this is what you need to know. It's a lot of information there, but most psychiatric medications operate by this action. This explains why these types of meds require several weeks of administration. So psychopharm meds require several weeks of administration before symptoms are improved or there's a noticeable change. So we'll talk about antidepressants when we get to antidepressants and how long it can take those medications, a minimum of 4 to 6 weeks to notice a difference. Here are your major transmitters. So I'm sure you've heard of some of these, such as your catecholamines. These are based on chemical structure. So dopamine, serotonin, and norepinephrine. Your amino acids are GABA or gamma aminobutyric acid, glutamate, or glycine. And you'll notice here GABA is inhibitory and glutamate is excitatory, and that's important to know moving forward. And acetylcholine. Next is a little table that I created that breaks down your neurotransmitter, which you'll see on your left-hand side, and any implications or roles it's responsible for. So up here you'll see acetylcholine. In your CNS, it plays a role in memory, learning, attention, mood, rapid eye movement that occurs during sleep and behavioral arousal. In your PNS or peripheral nervous system, it is located at the synapses in which the nerve terminals meet the skeletal muscles. And this excitation results in those muscle contractions. It's important to know that in Alzheimer's disease, there is a lack or deficiency of acetylcholine in certain regions of the brain. Next is epinephrine or adrenaline. This is more active in the PNS. It is secreted by the adrenal glands and regulates our fight or flight response, along with norepinephrine, because they act on opposites, which we'll get to. Norepinephrine is synthesized from dopamine. It's primarily excitatory in the CNS. Norepinephrine cell bodies located in the brainstem have axons into the limbic system, which is your emotional epicenter, and your frontal lobes. It is also involved in alertness and wakefulness, which are two major functions, and it also plays a role in mood disorders. Dopamine is involved in behavioral regulation, learning, mood, attention, and movement. It may have excitatory or inhibitory effects. Cocaine, amphetamines, and other drugs are directly activate dopamine receptors, which may account for the psychotic symptoms that are observed with drug abuse. Overactivity of dopamine or oversensitivity of dopamine receptors are said to be the cause of schizophrenia symptoms, and that's the dopamine hypothesis. And then a decrease in dopamine is what can cause the muscle rigidity that is seen in Parkinson's. Serotonin, the precursor to that, is tryptophan. In your CNS, it is involved in the inhibition of behavior and activity, control of appetite, mood regulation, sleep, arousal, and pain regulation. Depression, impulse behavior, and aggressiveness also involves some serotonin imbalances. GABA, which is inhibitory, is involved in motor control and vision. Barbiturates and benzodiazepines increase GABA, so it increases inhibitory responses. It is used to treat epilepsy and with trembling that is associated with Huntington's disease. Glutamate is excitatory. This is associated with learning and memory, and it lowers the threshold of excitation for neurons. Glycine is inhibitory. It's mostly in the spinal cord where when receptors are activated, the cell hyperpolarizes and is less likely to fire or transmit a signal. And the information was taken from your basic pharm book. So here is the 7 steps in neurotransmitter action, which we've already talked about, and we're going to do this again because just repetition is going to help you understand this. And here it is labeled 1 through 7 here on the side, so it does help you with that. So neurotransmitter molecules are synthesized from precursors under the influence of enzymes. So just like tryptophan is the precursor to serotonin. And just like we talked about tyrosine being the precursor to dopamine. So neurotransmitter molecules are stored in these vesicles, which we should all understand that concept at this point. Neurotransmitter molecules that leak from their vesicles are destroyed by enzymes. Action potential causes the vesicles to fuse with a presynaptic membrane and release their neurotransmitter molecules in the synapse. So as you can see here, this is that fusing of the cell. Release neurotransmitter molecules bind with autoreceptors and inhibit subsequent neurotransmitter release. 6, this is when released neurotransmitter molecules bind to the postsynaptic receptors. So in the previous, in number 5, it's saying the released neurotransmitters bind with autoreceptors and inhibit subsequent neurotransmitter release. So we'll talk about that more when we get into certain medications. And number 7, released neurotransmitter molecules are deactivated by either reuptake, so they go back up into that presynaptic membrane, or enzymes destroy those molecules here in the extracellular fluid. Next are mechanisms or ways that neuronal dysfunction can occur. This includes chronic stress of drugs, the inhibition of neurotransmitter synthesis, meaning there's too little, excessive degradation of neurotransmitters by enzymes, biologically based diseases that alter release, so too much or too little, abnormal reuptake by presynaptic neurons, abnormal up or down regulation of receptors, and alterations in gene expression and cellular processes by second messengers. So there are several common dysfunctions that are believed to underlie major mental illnesses. One of these being the initial synthesis or production of neurotransmitters may be inhibited So this results in too little of a neurotransmitter being available to be released. Neurotransmitters may be subjected to excessive degradation by enzymes, such as MAOs, which are monoamine oxidase, and anything that ends in that ACE is an enzyme. Certain biologically based disorders or drugs may either facilitate or inhibit the release of neurotransmitters by the presynaptic bouton. Another way is the process of reuptake absorption may be altered. So an example of this would be cocaine abuse leads to blocking of dopamine reuptake pores. In clinical major depression, reuptake of serotonin, norepinephrine, and or dopamine may be accelerated. So those are two examples of how the reuptake absorption may be altered, which results in neuronal malfunction, which is believed to be an underlying reason or occurrence for major mental illness. Another common dysfunction is receptors may be either abnormally upregulated or downregulated. Or pathological alterations can occur in gene expression due to marked changes in second messengers and may result in numerous abnormal cellular responses. This includes cell growth, altered receptor sensitivity, dendrite retraction, or loss of dendrite spines, or obviously cell death. So here is a visual that shows you, once again, the presynaptic and postsynaptic, but it also shows you different action types. So here it's showing that amphetamine and cocaine block the reuptake of dopamine, and that is an agonist, which we'll get into the terminology and definition of agonist, antagonist in a little bit, but it's basically what you already understand regarding those terms. Amphetamine promotes the release of dopamine. So it blocks the reuptake and it also promotes the release. And then an antagonist, such as Thorazine, which is used to treat schizophrenia, psychotic disorders, and mania and bipolar. Here it shows that it occupies the dopamine site on the D2 receptor, which prevents the receptor being activated. So here are the descriptions I said that we would be covering. So excitatory means it increases activity or it's depolarizing. Inhibitory is it decreases activity or hyperpolarizes. An agonist increases the effectiveness of neurotransmission, so it excites or aids. It can mimic neurotransmitters. An example of this being that heroin mimics endorphins. And then you also can have a partial agonist, so it activates the receptor but to a lesser degree. So an example of this would be Suboxone, which is a medication that is used to help with those that are addicted to narcotics or opioids. An antagonist, this decreases the effectiveness of neurotransmission. It inhibits or blocks. An example of this would be naltrexone, which attaches to the opioid receptor without actually activating it. So by attaching to it and it's not activating it, it blocks any additional opiates from going and binding to the receptors, causing that chemical reaction. Next is synaptic pruning, and I want to talk about this because we will talk about this in later modules. Synaptic pruning occurs mostly in adolescence and childhood. It is when unnecessary neuronal structures are removed by 50%. Which allows for them to be replaced with more efficient and complex structures. So this is what it's more beneficial in adulthood to have the synaptic pruning take place. And there are some disorders that are thought to be the results of dysfunction or non-efficient synaptic pruning that took place in childhood and adolescence. Brain structures, going way back into biology. So for practical purposes, the brain is divided into 3 basic units. You have your brain stem, you have your central core, which is your limbic system, diencephalon, and basal ganglia, and then you have your cerebral cortex, which is the highest area of the brain, and this is the area that is responsible for perception, complex cognitive processes, reality testing, and initiation of behavior and judgment. So your major mental illnesses is said to involve neurochemical dysfunctions in various subcortical areas, such as your limbic system, which I briefly spoke about as being that emotional epicenter. Basal ganglia. This area is important for movement. Damage to this area results in Parkinson's and Huntington's. This area is critical in learning and remembering how to do things, planning, attention, and language. Structures that are contained within the basal ganglia is the caudate nucleus, the putamen, and the globus pallidus. Also, there is the reticular system and the brainstem, all of which play critical roles in mediating primary affective states in a host of biological rhythms and drives such as sleep, sex, and hunger. And brain function can be derailed or damaged, or any damage to the brain tissue by trauma, toxins, or disease, or by malfunction occurring in the nerve cell. There is a visual of those brain structures that we just spoke about. You can see your amygdala over here in your hippocampus, and here's the putamen and the globus pallidus and the caudate nucleus that you'll see in your basal ganglia. Back up here to the limbic system is your amygdala, your hippocampus, which we'll talk about, your cingulate, and your septum. There it is. It's really tiny. So your hypothalamus, this is the primitive brain center. It is actually the size of a pea, and it is located at the base of the brain. It is the complex structure that regulates biological functions, such as circadian rhythm, sleep cycles, appetite, sex drives, and the regulation of both the autonomic nervous system and almost all of your endocrine glands. Neurotransmitters, serotonin, norepinephrine, dopamine are thought to be crucial in regulating certain aspects of hypothalamic functioning. And damage to this area affects motivated behaviors such as drinking, temperature, feeding, sexual behavior, activity level, and fighting. The limbic system. So this is that emotional brain center I was talking about. So the structures within the limbic system are the amygdala, the septum, the cingulate gyrus, and the hippocampus, which is responsible for memory. Damage to this area results in difficulty storing new memories. And the way I kind of explain hippocampus to help people remember, there's different ways to assist in memorizing the hippocampus. So one of them is campus, so it is the campus for your memory. That's one way to remember it. But another way I like to remember it is it says the word hippo. So whenever this is a crazy way to remember it, but this is something that back when I was learning this years and years and years ago, have you ever heard of, you have the memory of an elephant. So this is, you have the memory of a hippo. So it's just a fun way to remember the different areas of the brain. And the limbic system is involved in 3 primary functions. This includes the appraisal of emotional stimuli, the initiation of emotional responses, such as seen with fight or flight, and shutting down reactivity after external stressors subside. This is actually found in your book, and it shows you the brain structure, the functions, and any psychopathology that is associated with those specific brain structures in which the functions are damaged. Information processing. So incoming sensory information is appraised by way of 2 main processing centers. The cerebral cortex, which is the most highly evolved part of the brain. It is capable of complex information processing and critical thinking. Next is the amygdala. You actually have 2 amygdalae, one in each temporal lobe, just as an FYI. But the amygdala has the high capacity to register, perceive, and analyze sensory data. But it also can result in poor appraisal of environmental stressors. It is able to engage in gross pattern recognition. If such patterns resemble objects or events previously associated with danger, your neuronal impulses exiting the amygdala elicit a fight or flight response. So that is something like if you have, which results in lots of false alarms. It's rapid and inexact. So an example of this would be if you are deathly afraid of snakes and you're walking and you see a garden hose or a stick on the ground that resembles a snake, your appraisal of that situation is you see a snake and your body will react in the way if it was a snake, even though it's a stick. This just shows the divisions of the central nervous system, your CNS, your PNS, and the reason why I wanted to show this is it shows your autonomic and how it's broken into sympathetic and parasympathetic, which is involved in your fight or flight response. So when a person is confronted with a stressor, it is interpreted on two levels, and then an emotional response is initiated, and that is your cortical and subcortical. The main pathway for this involves neuronal activation of the hypothalamus by the amygdala. So we just talked about what the role of the amygdala is. So it tells you what is the perceived danger. So at the hypothalamus, there's three primary stress response pathways that are then activated. The first and most immediate felt response to the perceived danger is evoked by the sympathetic nervous system. Your SNS nerves travel to almost all areas of the body and release norepinephrine, which activates adrenergic receptors and cause increased heart rate, blood pressure, and sweating. So that is your fight or flight response kicking in. Your SNS nerves also penetrate the adrenal gland in the adrenal medulla, which then discharges norepinephrine and adrenaline into the bloodstream. Your neuroendocrine pathways, which is a brain activation of endocrine glands. You have your hypothalamic pituitary adrenal pathway, or it is also referred to as your HPA axis. It results in the release of steroid hormones from the adrenal gland, which includes hormones such as glucocorticoids, which facilitate the release of glucose into the circulation. Humans' main stress hormone is cortisol, which I'm sure you guys have seen and heard of cortisol. It is a stress hormone. Your HPA axis, when it is activated, it causes the release of two thyroid hormones, T4 and T3, and to quickly mobilize the body to take action to avoid danger. So that's your fight or flight response. So that's when you're taking action to avoid danger. So you're either attacking or you're running. Returning to baseline. So the various stress hormones re-enter the brain and trigger a shutting down of the stress response. So that's after your body and your brain has realized that you are not in danger. The hormonal feedback allows the nervous system to regulate itself, and when danger is no longer a threat, it returns to homeostasis. Your hippocampus. This is limbic structures that reduce arousal. It turns off stress response, and it shuts off that HPA axis. Your hippocampus is densely populated with glucocorticoid receptors, also known as GR. When cortisol molecules reach the hippocampus and bind to those GRs, that activates the hippocampus, which then exerts inhibitory control over the hypothalamus and thus deactivates the stress response. Some research suggests that the anterior cingulate gyrus also plays a role in reducing emotional arousal. Your PNS is a somatic division. It controls voluntary action of the skeletal muscles and carries information from sensory organs to the central nervous system. A second branch of the PNS is the autonomic nervous system, or your ANS. Here you have involuntary organs such as your heart and smooth muscles and glands. Your ANS breaks down the sympathetic and parasympathetic systems. Your CNS plays a role in the fight or flight response. It is set in motion by activity in the limbic system and hypothalamus. The SNS mobilizes the body to take action in response to dangerous situations. The parasympathetic system is activated at times of relaxation. It acts to reduce the heart rate and blood pressure and overall attempt to conserve energy. So here is your fight or flight response and how it affects your anatomy. So your sympathetic division, which is over here on your right-hand side, this dilates the pupil. It has no effect to tear glands. It accelerates your heart. It constricts arteries. It dilates your bronchi. It inhibits stomach motility and secretion, inhibits pancreas and adrenals. It inhibits intestinal motility, relaxes bladder, and it stimulates ejaculation. Your parasympathetic division constricts pupils, it stimulates your tear glands, It inhibits heart, it dilates arteries, so it does the opposite. It constricts bronchi, it stimulates stomach motility and secretion. It contracts the bladder and it stimulates erection. Psychotropic medication and the parasympathetic and sympathetic nervous system. Psychotropic medications can affect parasympathetic and sympathetic nervous systems, which produces side effects. The sympathetic system is mediated primarily by norepinephrine. Certain psychotropic medications either block or activate adrenergic neurons in the sympathetic nervous system, producing those undesirable side effects. So this is actually that top part right there and bottom part right here. That is actually what we saw on the visual of the body. And lastly, here's all the sources that I used to get all this information. You guys did it. I know it was a lot of information, but that is, once again, that is why I put so much text within each slide, because I didn't want you to be feverishly trying to take notes and missing things. So I want you to be able to just refer back to this. And next we'll be moving on to the next module.